Humoral immunity and cell-mediated immunity. Humoral immunity and cell-mediated immunity are the two types of an adaptive immune response that enable the human body to defend itself in a targeted way against harmful agents such as bacteria, viruses, and toxins. Humoral immunity. Humoral response is primarily driven by B-cell lymphocytes that produces antibodies against foreign antigens, which are freely floating in the bloodstream or outside the infected cells. Hence, it is also known as antibody-mediated immunity. It is so named because humor refers to the body fluids, generally blood, where the antibodies circulate freely and bind to the specific antigens, neutralize them, and causes lysis or phagocytosis. This is a macrophage, which is a phagocytic cell. In humoral response, the antibodies are secreted by the plasma B cells. These antibodies bind to the specific molecules called antigens present on the surface of the bacterium, thereby neutralizing the pathogen. This neutralization prevents the entry of the pathogen and bacterial toxins into the cells. The antibody-caught pathogens bind to the receptors on macrophages, which then phagocytose the bacterium and destroys it. The second type is cell-mediated immunity. Cellular immunity occurs inside the infected cells, and is mainly mediated by T-cell lymphocytes of the immune system. Hence, it is also known as cell-mediated immunity. It involves the activation of antigen-specific cells, such as cytotoxic T lymphocytes and macrophages, which recognizes and destroys the infected cells harboring antigen. It primarily defends against intracellular pathogens, multicellular parasites, transplanted tissue, and cancer cells. T helper cells and cytotoxic T cells are the key effectors of this cellular response. In cellular response, the process begins when a naive TC cell encounters an antigen. The CD8 plus TC cell with T cell receptor recognize the peptides presented by class I MHC, found on the surface of the antigen-presenting cells, such as dendritic cell. The activated cytotoxic T cells further proliferate and differentiates into armed effector cells that destroy the infected cells by inducing apoptosis. The memory TC cells are differentiated T cells that are activated upon re-encountering the same antigen. This picture represents the mechanisms of both humoral immunity and cell-mediated immunity. When antigens, such as foreign protein, virus, and bacteria, invade the host body, humoral response begins with the activation of the B cells. Binding of foreign antigen to the B cell receptors promotes the differentiation process that results in the formation of effector B cells, which are the antibody-secreting plasma cells that eliminate the pathogen. Memory B cells are also formed in this process. Cellular response begins when T cell with T cell receptor recognizes the foreign antigens. The activated TC lymphocytes further differentiate into TC cells and T helper cells. The cytotoxic T cells cause lysis of the pathogen-infected cells. On the other hand, T helper cells recognize the antigens bound to class II MHC molecules and release cytokines. These cytokines further promote the activation of B cells and T cells. With assistance from T helper cells, the activated memory B cells differentiate into plasma B cells that can produce antibodies against a specific antigen. This summarizes the comparison between the mechanisms of humoral immunity and cell-mediated immunity. Humoral response begins when naive B cells encounters a foreign antigen. Binding of foreign antigen to the B cell receptors promotes the differentiation process that results in the formation of effector B cells, which are the plasma cells, and memory B cells. With assistance from T helper cells, the activated memory B cells differentiate into plasma B cells that can produce antibodies against a specific antigen. Cellular response begins when T cell recognizes the foreign antigens on antigen-presenting cells. The activated TC lymphocytes further differentiate into cytotoxic T cells and T helper cells. The cytotoxic T cells cause lysis of the pathogen-infected cells. On the other hand, T helper cells recognize the antigens bound to class II MHC molecules and release cytokines, which further promote the activation of B cells and T cells. Thank you for watching. Please do like and share this video. Also, click on the subscribe button and hit on the bell icon to get the notification of our latest videos.